here on this platform, we are giving a brief idea and brief knowledge about the topic of hemoglobinuria in sheep and goat, the causes and diagnosis. This is one of the plant toxicosis where you find hemoglobinuria. So in India and in many regions of the world where cotton is being produced, the cotton seeds, they are used as oil cakes for animals. They are used as source of protein when the crude oil is extracted, and that is used for some cosmetics and some medicinal industries. But when the oil is extracted and the remaining portion is there, it is rich in protein, so it can be used as concentrate, and it is used as concentrate in many areas. So gossypol, the cotton plant, it has glands, and the if the sheep and goat, they are let loose to graze on these plants, or if the cotton cake is being fed in huge amount to the animals, then there is toxicity. Usually this is toxic in case of animals in sheep ruminants, when the rumen is not properly developed. Okay, in case of young animals, so this is more common. So after consumption of the cotton seed, there is excess amount of free gossypol in the blood, and then that will cause hypertoxicity in the hyper amount of gossypol in the blood, and that will lead to toxicity. Now what happens, the affected animal, there will be hematologic changes, and again there will be increased RBC fragility, that will lead to lysis of the RBC, that will lead to anemia, that will lead to icterus. And again, the gossypol also has some effect on the heart tissue, so there will be cardiac failure and sudden death. This will also get absorbed in the liver, and there will be liver necrosis, and there will be reproductive failure, because since there is anemia, there is cardiac failure, there is cardiac affection, there is liver affection. So all these things will lead to reduced circulation in the deeper part of the tissues in the body, and also that will cause, because there is lysis of the RBCs, again there will be anemia, and all these things will lead to reproductive failure in case of animals. And along with that, certain researchers have shown that it affects the spermatogenesis and libido in case of males, and in case of females, there will be irregular cycling, there will be irregular cycle of ovulation, and if at all there is pregnancy, there will be embryonic death, and there may be abortion. So the only treatment here we suggest is removal of the cause, that is removal of the gossypol from the feed, and give some supportive therapy. So how to diagnose this? So again, a history that the animal was exposed to the cottonseed meal, or the animal is being fed for a long time on cottonseed meal or products, and then the clinical signs, that is there will be sudden death and anemia and all, and then there will be reproductive failure. And again, if you see these steps, and many times in the field we do not even go for laboratory diagnosis uh, because of unavailability of facility, we don't go for diagnosis, we simply see the symptoms and we go for treatment. So most of the time we are treating with antimicrobials and supportive therapy. So one differential diagnosis here can be that, like if the animals are being treated with antimicrobials and still the animals are not recovering, the animals are not showing proper growth and all, so you can suspect that there is some phytotoxic effect, and there is some plant or plant product which is causing toxicity. Then post-mortem you can find that there is cardiomyopathy and hepatopathy, and there will be tissue fluid accumulation. And along with that, if you go for sending the tissue samples to the toxicology laboratory, you can see that there is increased concentration of free gossipol in the tissues. And also if you go for analysis of the feed, you can also find high concentration of gossipol in the feed or in the diet of the animal. Then how to go for differential diagnosis? So complete blood count is to be seen. If there is reduced amount of RBCs and other things like WBCs and all, they are normal, then we can suspect that there is some non-infectious cause. Because in infectious cause, definitely the WBC picture is going to be high. Then you can also go for blood picture examination, blood slide preparation, and then staining the slide. So you can go for identification of some intraerythrocytic protozoan parasitic infection if it is present, or there are some toxic bodies present in the erythrocyte or remnants in the erythrocytes. So you can go for identification of that. Then you can also discriminate whether this is a regenerative anemia or non-regenerative anemia. If you find more number of reticulocytes on the smear, then you can definitely say that this is a regenerative anemia. Then a serum biochemistry panel can also be tried. You can go for liver function test, then kidney function test. Then you can go for estimation of the different elements in the liver and tissue, especially as I said that these are the important causes of anemia, hemolytic anemia. So you can definitely go for estimation of nitrates and estimation of your copper, molybdenum and all. This way you can find out the trigger of hemolytic anemia. Then plasma hemoglobin concentration, that is definitely increased. And because of that there will be because of intravascular hemolysis, the plasma hemoglobin concentration will be increased. Then serum LDH concentration, that will also be increased because of damage to the kidney, heart and liver. So there will be splenomegaly. If there is splenomegaly and there is absence of intravascular hemolysis, and there is presence of extravascular hemolysis, then the cause should be no. Extravascular hemolysis, there will not be splenomegaly, and if there is intravascular hemolysis, there will be splenomegaly. Then you can also go for dipstick test. Urine dipsticks are available, and you can just take the fresh urine, midstream urine you have to collect, and then you can dip the test strip, and you can check the color change, and you can correlate with the colors on the particular dipstick box. So they give a color index, and accordingly correspondingly you can go for checking of that, and then you can find out whether it is a hemoglobinuria, whether it is a myoglobinuria, hematuria, or some other reason which is causing urine to oxidize, and thus it will cause later on when this urine is kept standing for a while, for 15 minutes or half an hour, the urine will change color from yellow to dark yellow, and sometimes that is very dark, that it seems like it is a hemoglobinuria. So iodine, bleach and all, they are to be checked. Then how to differentiate between hemoglobinuria, myoglobinuria? For this you evaluate the plasma color, then you go for hemoglobin urea.